Our next lecture is going to focus on exactly how we want to sample and sort of figure out if we can get to a local minimum in our energy landscape. And so what we're going to do is we're going to talk about what's a good searching strategy in general through the energy landscape and how exactly we can minimize our proteins. We partially covered this, and you've already seen this figure right here. So we talked about, in terms of this energy landscape and having sort of an unfolded protein and going all the way into very well folded, low energy, possibly the lowest energy state, and it might be the native, such that this is a naturally occurring protein in its lowest energy conformation, or perhaps we're sort of starting from scratch and making a de novo protein, and we're interested in figuring out exactly how to get it to the lowest energy conformation that we can. And so with Anfinsen's dogma, some things to keep in mind in general about protein folding is in experiments as well, so the temperature as well as solvent concentrations in general, what influence the folding event, and also when we have a native structure, which could just be the lowest energy state in general, we assume that this is unique, stable, and kinetically accessible minimum. So in terms of the free energy landscape, we assume that the native state is all of these things. So can you think of an exception to this? Is it possible to find a native protein in that it occurs naturally? But when we take a look at it, perhaps it isn't necessarily all of these things. So maybe the structure isn't necessarily stable. Are there any naturally occurring proteins that aren't necessarily stable? Yeah. So perhaps there are intermediate states, and maybe that sort of corresponds to not being able to figure out exactly how we can minimize through the energy. So that would be high up somewhere there. Anybody else? So maybe there are proteins that don't necessarily have well-defined tertiary structure, and maybe that is possibly related to their function. So they're not well folded in their lowest energy state all the time, especially maybe to remain functional. Anybody else? So we can have proteins that are misfolded and sort of influence the misfolding of other proteins. So these, and the formal name is intrinsically disordered proteins, they don't necessarily follow this logic. It's not that every time you can sort of reach a local minimum that is functional and is the native structure. Also, sometimes this occurs with just natural enzymes. For their function, certain sort of regions of the protein actually remain unfolded or very, very loopy, and that allows them to bind and be flexible and sort of do everything that an enzyme has to do. So given that this search is made difficult by these exceptions, we're just going to focus on sort of the types of proteins that we assume have a lowest energy conformation, and that we can either get close to searching for it or find it or be inspired by some natural examples. And in this case, of course, we're doing physics-based modeling, and in order to find the structure of a protein, we're going to try to get to that energy minimum. And this is very rough pseudocode to sort of communicate the idea is if we're interested in predicting the structure of a protein given its amino acid sequence, we want to return minimum of all sampled conformations, and the criteria of this minimization would be free energy. So you want to get to the lowest possible free energy given all conformations. Ideally, you would do every single one, so every single possibility, but we know that that is not a very easy task. We know that we don't have infinite time. And so in general, the idea is to predict and minimize on all conformations. And there's also other problems that sort of complicate this task. Is given a protein backbone, how do we know that we have the right sequence? And how do we know that we were able to sort of figure out what sequence would fold into the backbone structure that we're interested in? And so this itself is basically the protein folding problem. And with side chain design problem here, the idea is that the sequence can also change. And how do we know that we've sort of changed the sequence to the point that it doesn't look like the protein backbone that we started with? So you've sort of worked with point mutations earlier today, actually just now. And one question I have for you is it possible to have one point mutation break your entire protein structure? Is it possible to just mutate one residue and then change the entire protein fold? Either for it to be misfolded or for it to just look like a different topology. Raise your hand if you think that's possible. All right, somebody care to explain how or why? So maybe we mutated to a proline, which is going to break some structure. So it's possible to make a mutation such that the entire structure collapses, perhaps, and it looks nothing like what you started with. You can also think of this in regards to one specific residue, because we know with cysteines what is something that you can have. We have disulfide bonds, and what's special about disulfide bonds? Let's try to relate it to structure. So you saw in the energy terms for Rosetta that disulfide is calculated and it contributes to the overall energy. So it turns out that having these disulfide bonds almost staple the protein at certain residues. So you can have the 3D structure sort of come together and make a disulfide where it's very, very stable because of that bond. And sort of mutating one of the cysteines to something else would break the disulfide. So that could have many consequences in terms of the structure. And so in general, we're just going to talk about if we were to change the sequence given a backbone structure, perhaps this is a backbone that we've perturbed using some of our movers. So we would want to search for a sequence that will accommodate this backbone structure through all possible sequences. And if the backbone structure that we started with is similar to the predicted one based on the sequence, then we would accept this prediction and the sequence that we designed. And what is a little bit problematic about searching for all possible sequences? Let's focus on what is problematic about figuring out what all the sequences are. Sort of making something that contains all possible sequences. So it becomes too exponential too quick. And, you know, we covered this concept a few times. There is a particular idea here. What is the name of the... Right. And so, again, for the average length being 300 amino acids long, it becomes really problematic really, really quickly. So we're not going to be able to predict the structure for all sequences possible and then make the comparison to the backbone. And imagine if we're making perturbations to this backbone and it's not taken from the PDB. This is our design and we're sampling a bunch from a bunch of different movers. And let's say that we're interested in sort of figuring out a bunch of different sequences for this and see if they look similar at all. So then the problem becomes very hard to approach. And so using free energy, our goal is to have a little bit of a shortcut and also have some guidance and some bias to this process so that we're mutating in a way that is favorable in terms of energy, or we're modifying the pose in terms of its structure such that it's favorable in terms of free energy. And let's say that we have some type of conformational space and we're interested in sampling either sidechain or backbone coordinates. And our goal is to figure out what sort of movements we can have in those atoms such that the energy is minimized. And then we're also going to search the space of sequences and figure out for these conformations what's a possible sort of sequence that will give us the structure. And this is a combination of the sort of the two code snippets that you saw. So the idea is to search over all sequences and all possible but physically confined conformations based on free energy and then pick the minimum. So let's consider this in terms of minimization, where we know that there are different minimization algorithms and this exists in different fields. And so in Rosetta, we also want to sort of minimize and do this in a smart way so that we're efficiently searching through the backbone conformations that are possible to be beneficial. And in Rosetta, the minimization process, and we're going to talk about specific integrated protocols for minimization, it's deterministic. And so what does it mean to have a deterministic process? You reproduce the results every single time. And in terms of energy barriers, so to put this more pictorially, in search of our, let's say we're interested in this energy minimum. So we have energy minimum 1, we have 2, this is our free energy right here. And in the search, this specific sort of energy barrier can make it so that we end up thinking the entire energy landscape looks like this. So if you didn't search for the rest of it and didn't know that 2 existed, you would accept this to be the native state. So in minimization, it is a problem to sort of go through the energy barrier that can allow you to access a lower energy conformation or a lower energy state. And you can sort of see it in here. So different barriers, searching through this space. And in Rosetta, the idea is to sort of only go if you're able to descent, so that you're not trying to reach higher in energy, and you only accept mutations or perturbations in a certain direction. And you determine a step along the descent direction, and then choose to basically either repeat, and we're going to talk about exactly what influences or what parameters sort of go with this type of search so that you're guaranteed to go from state 1 and search, and then sort of minimize to state 2. Ignoring all of this for a second, do you think it's possible that we now have reached state 2, we've minimized at this point? Do you think we can ever figure out if there is even a lower energy state than state 2? Do we ever actually figure out that we have the absolute lowest conformation we can get for a given sequence? Can you think of an example where we reach state 2 and we know that it's done, and it's not the local minimum, it's the global minimum? So let's say that we've only searched up to there, and we don't know the full picture. So we don't know exactly if from this point onwards it goes up or down. So we're not going to sample through every possibility. So we don't have a continuous energy landscape through every possibility. But nonetheless, is it possible for us to know that we've reached a global minimum? So talking about temperature, so increasing temperature allows us to sort of jump energy barriers, and decreasing it, sort of freezing the proteins, let's say, allows us to sort of get to a point where we're reaching the minimum. But then the problem with that type of a search is that you never know if at state 2, if you did increase temperature, that you would make this energy barrier, and then once you clear that, you get to a state 3 that's even lower. So what if we, instead of having one starting point and then sort of searching to state 1 or state 2, we have a bunch of random starting points, and then we randomly search, and then we have an intelligent way to guide this search, and we have some way to sample. We have some influence of temperature, and then we end up somewhere that perhaps with some confidence, we're sure that we're at a global minimum. So we don't necessarily always have absolute answers, but we have very good solutions to biological challenges. So it's not always about for sure knowing that, okay, state 2 is the end all be all, but if state 2 is a very stable therapeutic, then maybe it clears for a clinical trial and it helps so many people. So this is sort of the power of computational sampling and scoring, and then doing things in this physics space and searching through this space. And also, we're going to talk about exactly how step size influences things in terms of your descent direction as well. So this is a way better figure of what I was just trying to draw. And as some of you pointed out, we have a few things we can observe here. So first of all, we can sort of see that we have multiple starting points. So maybe these were randomly chosen for us to sample. And then these proteins look slightly misfolded at the very least, if not entirely misfolded. And we know that this isn't their lowest energy conformation. And we're trying to make it through this energy landscape and search from multiple points and see if we can get to a very low energy conformation that looks very well folded, and perhaps there's a structure we can compare this to make sure it's possibly compared to that structure folded even better. Or it's folded to a higher stability conformation, which we can also check experimentally. And so when we looked at centroid versus full atom comparison in Rosetta energy, we talked about the free energy landscape being rugged. And this is true of other types of sort of macromolecular assemblies as well. So if you were to consider a protein bound to RNA and you had different starting points, that also would make its own energy landscape where the energies of multiple molecules are essentially coupled together. And there is more to search from in that scenario. So you can really complicate this energy landscape scheme really, really quickly. But our idea is to at least get close to this global minima and figure out some method that's able to jump energy barriers. So one example of this is you sort of get to a, let's see. So let's say you start here and you minimize a little bit, you get to this point where the energetics look better than the previous step. And then the next sampled conformation is right here. And after that, you can sort of fall into this trap. So this local minimum, it looks fine, but if you stopped at this point and didn't have sort of enough of a step size or enough temperature or other factors to clear above this energy barrier and make it into the global minimum, then you wouldn't be able to search through most of the space, and you would be missing sort of a lot of conformations that are more stable than the one that you're choosing. And any method that you design to address this should be able to go across free energy barriers. And we already know that the coupled sequence and structure space is huge for proteins. We talked about why this is, so you saw this on the first day. We also talked about exactly how long this would take, roughly, and we just knew that it would take forever and that we're not interested in doing this. We talked about Levinthal's paradox, and in here the idea is if you were to sort of sample every single possible state and then make a comparison, but that's also not possible. And so to answer all of this and have some approach and some method that actually makes sense and gets us to maybe a good answer, maybe it's not the perfect answer, but it's a good answer, is going to be a sampling with Monte Carlo. So Monte Carlo based methods essentially have random number of sort of, well, there is some randomness in terms of how you're sampling through the space, and we talked about having random points of entry to the energy barrier, and also when you're searching through, let's say, Rosetta energy landscape, you're going to have some parameter for temperature. So in this case, we have KT shown here as an argument for Monte Carlo sampling. Anybody want to tell us what KT is? So we have thermal energy that we're simulating in terms of KT. And with different values of KT, perhaps we want to vary it through our random search so that we're able to make it past this energy barrier. We also have something called an iteration. So number of iterations for what? Let's just first acknowledge that our initial state is some conformation that we're starting with, okay? And we know this is going to relate to sort of how much thermal energy is in the system or whatever we're simulating. And then we're going to talk about the overall logic through the pseudocode. So at our starting point, the state that we accept is always the initial state because there is no other state. And then for a number of iterations, so this might be the number of tries that we're willing to have, we can go through and try to sample a random state from the current state. So make some perturbations to the current state and randomly make those perturbations, and we'll call that our new state. And then calculate a free energy difference between our newly sampled state or the current state. So we're going to have free energy of new minus old. And based on that, we create some criterion. So in this case, we're going to accept or reject with some probability. And if we sort of sampling, okay, so we sample through this part to get a random state, and based on our random state that we accepted, if free energy difference is less than 0, which means what exactly? So we talked about mutant minus wild type. So DDG. So DDG being less than 0, is that favorable? That's good, okay. So once we reach a point where we think the new state is better than the current accepted state, then with some probability, we will accept our current state to be the new one. And we're going to talk about exactly how you can sort of sample and see how temperature influences the entire system and this acceptance criterion. There should be video on that. Okay, and so more formally, this KT parameter is going to be very important to our sampling, and we sort of put it in terms of proteins, why that would matter. But whenever you have a large sort of KT that's available, then you can overcome large energy barriers. When it is small, then you're able to converge to your local minima. So whenever you start sort of cooling down your system, it goes deeper and deeper into this well without moving above where local minima is, so it sort of converges to the exact right direction to get to the lowest state. And with simulated annealing, we're going to sample until the temperature is gradually decreased to the point we have converged to some conformation. What is a way to check that you converged on one conformation? So let's say that your entire method is built upon spitting out the final accepted conformation. And you want to see if for the final, let's say 5 iterations, you're converging on an answer. So one idea might be you can take all of the final sampled states and see how close they are to each other. So once you converge, the structures themselves start looking very similar to one another, because converging on an answer means you just have one unique answer and you kind of settle into that. And the opposite, so when KT is quite large and you're able to pass through energy barriers, you can also think of that as chaos. So you're sort of searching through anything, and you can go above any energy barrier to the point you can go all the way up to the sky. But with simulated annealing, and with some type of temperature schedule we can have and gradually decrease it, we're able to cool down to the point that we only reach the global minimum and just stop at that point. And here is said video. So, okay, so starting now, temperature is very high and we're gradually decreasing temperature until, in this case, our goal is to find the global maxima. And so you can just see visually, if this is sort of all there is to this landscape, that the global maxima is right there. And so in order for us to find this, we have to be able to make any energy barriers across over here. So increasing the temperature allows us to sort of search more chaotically until sort of we're guaranteed to go between possibly 2 local maxima until we converge on our final answer, which we know because sort of we cool down and settle into that one point. And if you sort of pay attention to what happens on this side, so in the very beginning, you're able to find this local maxima as well, but towards the very end, it doesn't move all that much. And at the very end, you're just sampling through these minor peaks. And also, to relate this back to our protein space, so if this was some type of full atom energy landscape and you were interested in coarsening it, you would be able to get rid of some of these very minor peaks such that it's quite a bit smoother, and then the search through, at least at the final converging steps, is faster and more efficient. And then once you have a set of sort of centroid-wise sampled conformations, you can actually calculate full atom energy and then make your selection based off of that. We're also going to have to talk a little bit about equilibrium in general. So we're interested in finding a state of our protein, and proteins exist in many states, and states of proteins, or just their conformations, are influenced obviously by temperature amongst many other factors. And let's say that we have our free energy and we're going to have some KT. We can calculate this for more complicated problems where the free energy depends on side chain repacking, or perhaps we're changing the entire sequence, or perhaps as it gets folded. So this is the example that we've already sort of analyzed, is that we're able to analyze free energy minimization in the context of protein folding. If we were interested in docking, then the idea is to minimize over the complex free energy, and you would have to consider exactly where the interface is. Do you think it would be possible for us to search for the exact region that makes the interface through minimization? So I have 2 proteins that are getting that that are in a complex, and we're going to dock with Rosetta. And let's just say that the interface is supposed to be like this. And let's say that I sampled a conformation that's docked like that. Do you think the free energy of the correct complex is lower than the wrongly docked conformation? Okay, so maybe we can sort of search for the correctly docked position of both of the proteins coming together and it having some lower Rosetta energy. Is it possible to have maybe two interfaces where there are two right answers? So can a protein have two interfaces for another, let's say, molecule? And would they be even in terms of the energy landscape? Is it possible that binding at site 1 influences the binding at site 2? What is that called? Allostery? What's an example of an allosteric protein? So hemoglobin would be a good example. So binding event in one site influences how the rest of the energy landscape is for the entire protein. And so you might want to think about what changes our approach based on all different types of problems you can couple on top of protein folding. So, almost a co-folding problem, or calculating for a collective assembly. And we briefly mentioned loop modeling and some of the challenges. And we also talked about starting from a backbone that you yourself designed. So there's no sort of native starting point. You're not taking a structure using backrub and just making some perturbations. But remember how you made, let's say, tripeptide? Instead of that, imagine using a bunch of Rosetta protocols and you make an entire protein. And using some approach, you're interested in figuring out if your de novo structure is possible to fold in experiments at all. And so what criteria would work for that? So with score function and different Rosetta protocols that sort of work to minimize the protein conformation, and we're going to talk about relaxing protein conformations a little bit. What you're interested in doing is going from some high temperature to some converged conformation through decreasing that temperature, and this can be generalized to sort of jump over any energy barriers until you get to the correct or close to the correct minima. And then once you do get there, don't lose the progress you made and converge on that answer. And we're going to talk about fast relax, and you'll get a chance to use it in lab. What happens is the score function that fast relax uses as criteria for acceptance or rejection for sort of sampling a new state is going to ramp up the repulsion score term. So we talked about Rev 15, and we know that there is some energy term that corresponds to just atomic repulsion. And with that, why would we be interested in ramping the repulsion score term at all? So if repulse or repulsion in general is highly punished or very, very disfavored, then you would sort of have your proteins, pretty, or certain residues pretty far apart from each other because they can't get close, right? That's upweight. So ramped to the point they're repulsed more than normal. And at what point in sort of minimizing a protein structure are you interested in everything becoming very, very repulsive? So let's go back to some figure. So in this case, let's just say this is our entire search. And over here, we're starting at very high temperature so that we're able to sort of make all of these jumps as needed and finally, hopefully, converge here. So as we're searching through and we have some misfolded proteins, do you think that the repulsion energy term is really high for proteins that are misfolded? the loopy regions, they're not very structure organized or together, right? They're already far apart. So is there a point to punishing by ramping up the repulsion term? Not really, but as we sort of go down and converge on an answer, one effect that we could see is you could sort of compress the protein into a rock. So if you were to minimize all of the energies and there wasn't necessarily a way to keep track of repulsion, you could end up somewhere that is also very unrealistic and everything is very close and compact together in the name of it being fully folded. But in real proteins, we know there are loopy regions. There's always going to be loops. There are loops in enzymes. There are functional loops. Sometimes the loop is sort of the signaling region. And so we're not necessarily interested in destroying anything that is slightly unstructured in our conformation. We're interested in reaching a point that is acceptable and realistic. And so once we get to this converging step towards the very sort of final iterations of the states that we're sampling and hopefully finally converging on an answer, we ramp up the repulsion term so that the structure remains relatively realistic at the very end instead of having even more sort of unrealistic collapse. And that finalizes the search. Also, fast relax protocol is very commonly used with not only just sort of one type of starting or maximum temperature and then you go all the way to, let's say, 0, but sometimes the temperature schedule itself is inspired by a distribution. So what distribution can you think of for sampling the KT going into your sampling algorithm? So one example is you can just uniformly sample, and let's say we're going from 1 all the way to 0 in 100 iterations. So you can just evenly do that. What's another example of some distribution? Let's just say that in the initial stages, for a very large protein, you might be interested in staying in a very high temperature regime for a long time because it might be quite more difficult to fold, let's say, 1,000 residue long protein compared to 100 residue long. So perhaps you can have your temperature be inspired by your protein size, especially for these types of physics-based approaches, as you might be interested in changing temperature and adjusting it to the specific protein. And then could you also think of a case where you're using let's say the small molecule that is recognized by the protein in your entire search. So would you change the temperature for when the ligand is present versus absent? Do you think the energy landscape should be heavily influenced by sort of removing the binding target? Do you think it becomes smoother? Is there more noise if you have the ligand in your sampling? So we're going to discuss some of these ideas and try to sort of put it into actual code and see the results of what actually happens and how Rosetta uses, how Rosetta puts together some type of search and convergence based on ramping with repulsion. And we're going to move on to the lab for Monte Carlo.